Hello, my name's Martin Chilvers, field crops pathologist here at Michigan State University.
Hi, I'm Damon Smith, Extension Field Crops Pathologist at the University of Wisconsin-Madison. So Marty, we're tasked today to talk a little about mycotoxin management and silage. And what's interesting about this is, there's sort of a class of fungi that cause this problem, but we're learning that it's actually pretty diverse. Could you just tell us a little bit about, what we know about who actually causes the problem?
Yeah, absolutely, Damon. That's a good point. So we've been doing some research in this area and related areas in terms of ear molds as well for grain corn. I mean, the primary one, right, that we typically think about is Fusarium graminearum. And we think about that a lot, right, Damon, in small grains as well as corn. And there's certainly other species, and we'll show you a slide of that as well in a second. But Fusarium graminearum and other Fusaria species, part of the challenge with those is the mycotoxins that they produce. So deoxynivalenol, that vomitoxin that most people are familiar with. And then there's other toxins too, like zearalenone, that can have like an estrogenic or infertility and reproductive disorder effects. And as we're learning too, there's certainly a number of other mycotoxins that can be a problem. And there's certainly a lot when we go looking, isn't there?
Yeah. Yeah,
Yeah.
and what's also interesting is we're learning that it can kind of accumulate, these mycotoxins can accumulate kind of in all parts of the plant, right? Could you just talk a little bit about the what we know about the life cycle and the different phases of the
Yeah, absolutely. So again, back to the classic Fusarium graminearum, we typically think about that as, a pathogen of small grains, head scab on wheat, but also ear mold of corn. Um, but it's not just limited to graminearum. And graminearum can also infect the stalks, the crowns, and we get splash dispersal of those spores, collection in leaf nodes and infection. Plenty of opportunities during that lengthy growing season for the infection process. And I know, Damon, your lab's been doing a lot of work segmenting the ears from the stalks. Can you tell us a little bit about what you sort of found there?
Yeah, it's interesting. We started with brown midrib hybrids, and then we've also looked at non-brown midrib silage hybrids as well. And we can see
Mhm.
that depending on the year, we get different types of infection. We can either have an ear infection or a stalk or maybe even a root or crown infection. And we get differential accumulation of we've been sort of hyper focused on deoxynivalenol or DON in those studies, but we get differential accumulation of that particular mycotoxin depending on the year and where the infection process actually happened, which is pretty interesting and actually a little bit different than what we've seen like in wheat, Marty. Like
Yeah.
there's a pretty good correlation where you get accumulation of mycotoxin in the straw portion of the plant because you get leaching of the mycotoxin out of the head. So most of the infections happen in the head and wheat, and then it,
Mhm.
it gets pushed down into the straw. So kind of a different process in corn where it just sort of depends on the environment and where the infections actually happen and where we get the accumulation. Could you tell us a little about, some of the sampling and what you're learning about these species and stuff in Michigan?
Yeah, yeah. So again, this is just sort of backing out to the fusarium that are causing issues in wheat. Also, a little bit of root infection of soybean and dry bean.
Mm.
So Michaela Brunig did this really nice study where she collected a whole bunch of samples of fusarium from these different crop species, wheat and corn. And we found quite a bit of diversity. So this is just sort of demonstrating, where the samples came from. And if you go to the next slide, this is showing you the diversity that was present when she looked at which species of Fusarium are there. So on that left-hand figure there, you're looking at what was present in the wheat. So you
Mhm.
can see that most of those squares are green, and that green indicates Fusarium graminearum. And the smallest square equals like one field sample, one particular isolate. So each square is a different field or sample set. But you look to corn and popcorn underneath it, which are essentially the same thing, there's a lot of different colors in there. And so this is just showing us that there's a lot of different Fusarium species out there causing infection on corn. Again, this is restricted to looking primarily at samples of grain. And so this is going to be reflective of silage, but we're also going to, if we were to do this on silage, there may be some other differences there with respect to some of these species having preference for stalk infection as opposed to grain infection.
Yeah, we're actually working on a similar study, Marty, where
Hmm.
we've been looking at corn, and it's interesting. We see a similar trend where there's a lot more diversity in isolates or species within the corn side of things than we find generally with wheat, right? So it's
Yeah,
kind of interesting, and we're not sure. I think some of it might have to do with the fact that just when the weather sort of lines up with flowering period in wheat versus when we have silking in corn, right? That could be driving
yeah,
that along with diversity in crops and cropping systems and rotation and that sort of thing.
yeah.
So could you tell us a little about just mycotoxin management and what you guys been doing with some integrated management strategies in Michigan?
Yeah, absolutely. So there's been a couple of graduate students here at Michigan State, a couple of those under Manny Singh, our agronomist, and our entomologist, Chris Defonso, has been participating in those projects as well. This particular couple of slide decks is from grain corn, but the message applies to silage as well.
Mhm.
And so in this particular study, Caitlin was really interested in looking at ear rots and Western bean cutworm damage. And so, the classic plant disease triangle, us plant pathologists absolutely love. We've got to get those matching tattoos one day. But here we've added in insects, right? So as well as having your standard 3 points, we're thinking also about the insect element as it's affecting that ear rot component. And I guess we can also broaden this, right, to damage within the stalks and how that may introduce mycotoxins. Have you guys worked on any insect elements there within your silage work?
We have not yet, but it's crossed our minds that that could be
Mm,
contributing here. And of course, we do see similar trends with, where we get insect feeding on the ears. We see the same thing like you in Michigan, where, we'll tend to see higher levels of ear rots in those cases, right? So
yeah,
definitely some linkages there. And thinking along the lines, if you think about this triangle, maybe, that insect feeding increasing the susceptibility of the host component, right? Of that.
yeah.
So, yeah, kind of interesting how all these things sort of interact, and
Mhm.
definitely some researchable things going on there with the insect side of things. So, what'd you guys find out of that work?
Yeah, so the main takeaway here is that essentially just using a trait to manage that Western bean cutworm. So the VIP3A trait, that Viptera trait in this particular example, was able to manage the amount of Western bean cutworm, both incidence, so the number of ears that were damaged, and then the amount of severity of that damage on those ears compared to other cry genes
Mhm.
that are available.
Yeah,
Um,
interesting. Yeah, yeah.
yeah, and
So,
then
yeah,
that
having that protection there really helps reduce that feeding. Thereby, we don't get that wound where those spores can actually enter, right?
absolutely,
Yeah.
yeah. And so that damage was what we were finding. So that ear rot index that's shown here on that x-axis is really, you could probably almost think about it as being that ear feeding damage index as well. I don't think we could really pull those two apart in this particular example. But of course, we get, as Damon just said, as we get damage on those ears, we get, you know, those fungal infections, and then of course that DON accumulation. So we see a pretty good correlation between ear damage from those insects and the mycotoxin accumulation. So the take home here was that it's a component of management. Um, some people got kind of excited about this. I'm like, whoa, whoa, whoa! Like this is yes, this is a thing, but it's not the entire disease story. Just managing the insects isn't going to make mycotoxins go away. Again, it's definitely an important part.
Yeah, it's sort of that continuum I sort of think of a continuum of resistance or tolerance, right? So like that insect feeding probably just increases the relative susceptibility of a particular hybrid, right? There's still other mechanisms of partial resistance at play here as well, right?
Exactly. Yeah. So I just want to back out again and another component of that plant disease triangle, of course, is the environment, what weather conditions are occurring. And I don't know how 2023 worked out for you, but it was really bad here in Michigan, Indiana, Ohio, in terms of the amount of mycotoxin in grain samples and silage as well. And so we just had a lot of moisture and favorable temperature conditions, during silking. The other thing that was really interesting in 2023, we had a lot of very uneven fields. So we had extended silking windows, plants at different growth stages. So it increased that risk window as well. So it wasn't, just 7 days of green silk. Now it's maybe 14 or 20 days of silking where, the plants are susceptible to that silk channel infection process.
Yeah, it looks like you guys were pretty wet in July there. We had some spotty cases. I wouldn't say we had as widespread of an issue like you did in 23.
Mm.
So certainly the environment really drives a lot of this, I think, and I sort of assume that we always have the host and the pathogen together. Like we have plenty of Fusarium in the Midwest, right? And so like the
Mhm.
thing that always is driving whether we have it or not is really this environmental component, right?
Absolutely. Environment can trump all at times.
Yeah,
Yeah.
yeah, yeah.
Yeah, and this brings us to risk prediction models, and I'll pick your brain in a second on this as well. But this is the Fusarium head scab model that our counterparts put together for wheat, and thinking about fungicide timing there. So May 31st, we had very little mycotoxin in our wheat because we were flowering around the end of May, and the model was showing very low risk. But as we moved through the end of that season, now we're looking at July 10th. The risk changed pretty dramatically, and I've been asking the wheat people to please leave this model open, right? It turns off as that small grain crop move out of flowering. So I've asked them, like, please just we appreciate any tools to check for risk, right, Damon? And you
Yeah.
know all about that. And so yeah, this is one model that I tend to look at. Unfortunately, it does start to turn off as we still may be silking in corn, but it's a helpful indicator. Again, it's built around primarily Fusarium, graminearum in wheat systems, but again, that is one of the species we know can infect our corn plants.
Yeah, I mean, these two slides are really a good visual, I think, Marty. In Wisconsin, the last couple of years, we've had fairly low level. I mean, we have had some Don and some Fusarium head blight damage in winter wheat, but spring wheat has gotten hit really hard here in
Mm.
Wisconsin, and I think a lot of it is due to the fact that when winter wheat flowers we've been kind of in this. I'm going to just back up a slide. We've been in this scenario, right? Like,
Yeah.
low risk, the temperatures just aren't there. We don't have quite the right moisture. But then when spring wheat is flowering, this is the scenario, right? Like
Yes.
it's much warmer, much more
Mhm.
conducive. This past season, our spring wheat just got absolutely hammered with Fusarium head,
Yeah.
So I think it really depends on how everything lines up to those narrow flowering windows. And I think tools like this can really help both in wheat and in corn as well. Yeah, you can see that risk there just as you move through the season really
Yeah.
ramp up. Yeah, yeah.
That's right. I put this slide in too. So this is a pretty new tool, and you can tell us a bit more about it. But this is at the Crop Protection Network, where Damon's been instrumental in this, but we're trying to basically set up a one-stop shop so we can go and check different disease models. So, Damon, can you tell us a bit about this and what Pierce has been doing here?
Yeah, so Pierce has been looking at kind of right around the silk window and trying to tie certain environmental components that really drive mycotoxin presence later in the season at harvest. So it's a little different way of thinking about this, right? So you get to the end of the season and you can measure mycotoxin, but really what's going on is it's the level of mycotoxin at the end of the season is driven by how much infection you have during that kind of silk window. And so the modeling that they've been doing is really hyper focused around that kind of risk period, and you can kind of see what we're doing here in these graphs. We set this risk period up, and this is really what we would deduce would be the silking window at this particular location. And you can kind of see here what we're looking at is whether that environment has been favorable for mycotoxin accumulation actually at the end of the season, right? So it's kind of
Yeah.
a different way of thinking about it, but it really has to do with what's going on at that silk time. So, what's interesting about this process, both in corn and wheat, is that the opportunity for the fungus, that window is actually quite narrow, right? So in wheat, it's maybe 7 or 10 days, right around the start of anthesis, right, or flowering, and then with corn, it's really just during white silk stage, right?
Hmm.
There are some other things at play, like if you have uneven fields,
Hmm.
and wheat, if you have secondary tillers that flower later, that can open that window up. But really, what you want to do is use these tools to really make that decision, during that flowering period in both of these crops, whether you actually need that fungicide to essentially protect the silks in this case, right? And then subsequently reduce the level of mycotoxins later in the season.
Yeah. I pulled this slide in too because I just thought it's a really neat thing that Ontario does. And so during the end of September every year, they go and collect ear samples from across the province and then test them for mycotoxins. So it's a really sort of good heads up for them as to what mycotoxin levels they're expecting to see.
Hmm.
And I found it really interesting to look at this over years. And I put this graph together. It's hard to like look at these in table format. So I think this is maybe a slightly helpful way to look at that. So just color coding it by every year, how much mycotoxin they're seeing. So green is less than half parts per million, so really not much of a concern. That yellow, and especially the red, and then that really dark red. That dark red. Now we're really
Yeah,
concerned. It's
that's
greater than greater than 5 parts per million. And
yeah.
this is probably very reflective of what is, this is not silage, but it's probably very reflective of what is generally happening in our silage fields as well. And I just highlighted a couple of really problematic years. And I think we struggle with this in terms of sometimes getting funding, right? Like it's not a problem this year. Like, it hasn't been a problem for a while. Well, it'll probably be a problem again. It's just a matter
Right.
of time,
Yeah.
unfortunately.
Yeah.
You can see 2016, 18, 23, really bad years. I think that their data from 2025, Albert sent me a slide. It looks very similar to 2024. Pretty minimal damage that season.
Yeah. Yeah. So now just kind of moving away from the environmental side of things, Marty, and just kind of looking at some of the silage management work you guys been doing. You want to talk a little about what you've been doing there?
Yeah, so really quick funny story. So we brought this guy on, Peyton, who's pictured there. Our dairy industry was really concerned about tar spot, and I think rightly so. We had fields that looked like they had been frosted or something completely dead,
Hmm.
cannot even be fermented, right? Too dry. So we brought him on as a graduate student, and we've been working on a number of different trials, looking at fungicide application timing, harvest timing, hybrid by fungicide, like you have, Damon, as well, and then some fungicide efficacy trials. So I'm just going to highlight a couple of those that we've been doing. So we have this is just generally the setup. We'll use a two-row chopper, four-row plots, 20-foot long plots that we'll go through and we'll spray fungicides, look at different hybrids and whatnot. And we've been using the same lab, you and I, so that we can compare our data in terms of quality and mycotoxin analyses.
So would you learn out of the timing trial here?
Yeah, I simplified this. This had fungicide timing component to it, but the fungicide we used, because we didn't have much foliar disease, that particular fungicide didn't do a lot on mycotoxins, and we'll talk about that in a second. But the
Right.
most important thing from here was just looking at harvest timing. So we aimed at early harvest, so high moisture, 70% moisture, an average, so 65% moisture, then a late harvest at 60%. So probably bracketing the early to late still may be acceptable, but looking at
Gotcha.
how that may affect things.
Yeah.
And as we might expect, and maybe a little bit surprisingly, the amount of mycotoxin did jump considerably the longer we waited for harvest. I need to go pull back and look at the number of weeks between these, actually, just thinking about it now. But it's somewhat expected, but maybe that response might not have been known before. I don't know, but I just thought that was kind of interesting that as we delay harvest, that mycotoxin level jumps. So I guess just thinking about things, this could be something of a strategy if we think we are at a slightly higher risk for mycotoxins. Maybe we should try and err on the slightly earlier harvest timing rather than letting it set. I do just want to point out too that the samples we take are collected immediately after chopping, but we dry those immediately. So we don't go through the fermentation process like you've been looking at, Damon, and that provides additional information that you've been looking at.
Yeah, yeah, we've done some mini silo trials here at UW, and we've kind of looked at this, and we continue to see mycotoxins accumulate even in the silo. So if you start with low levels going into the silo, you'll get incrementally higher levels coming back out. And we're working on the mechanisms of why that is. We think there's a couple of reasons for that. So if you end up with, like this later timing, Marty, this 60%,
Hmm.
now you got something that's almost a little bit too dry and it's
Right.
got higher mycotoxin levels, right? So that's like the worst case scenario.
Right.
Now you're going into the silo. You really can't pack the bunker correctly because you got a material that's almost too dry, and you got high mycotoxin levels that are going to continue to accumulate, right? So
Yeah.
I agree. I think one of the strategies here is just maybe if you think there's a problem there, is just go ahead and push that timing up just a little bit earlier, where you have a moisture level that can help you get that bunker to ferment quicker. Plus, you stand the chance of having just a bit lower mycotoxin going into the bunker itself.
Mm.
Yeah. So, what'd you find with zearalenone here?
So the same thing, Don and zearalenone pretty much sort of echo one another. Again, it's not one we typically test for, or most people might not be as aware of zearalenone, but it's the same thing.
Yeah. Yeah,
Yeah,
they kind of go hand in hand. Yeah.
yeah.
Yeah. And there's some synergistic things I've learned from our nutrition counterparts,
Mm.
right? So you get these interactions of these mycotoxins that we're still trying to learn about too, right? So,
Yeah,
sometimes the lower levels of both can be worse because probably some synergistic relationships as well.
yeah,
Yeah.
absolutely. And then just thinking about, okay, so what are the other quality components? Tonnage, milk production. Of course, we want to maximize that. So if we did harvest early, maybe we do give up something on the tonnage, but if you're going to sacrifice quality too much, it's just one of those things that we need to all be, paying attention to, I guess. And
Yeah. Yeah,
yeah,
you can have a lot of feed and you can have a lot of bad feed, and a lot of bad feed is not good, right? So,
that's right.
sometimes it's okay to give up a little on the tonnage side for a better quality product.
Exactly.
Yeah, so what about fungicides, Marty?
Yeah, and I know you've done a lot of work in this space as well. This is just again just a photo of some of the setup that we use, obviously spraying wheat and barley, but yeah, we were curious about fungicides.
Mhm.
I don't know how you feel about it, but I think it's been kind of frustrating to get good data on fungicides for mycotoxin management.
Yeah.
I threw this slide in from our colleague Carl Bradley at Kentucky, just the potential to maybe aggravate mycotoxin through strobilurin fungicides. And so this is a wheat trial. Again, we want to emphasize that this is not corn data, but it just demonstrates the potential um, so he's got four different locations here, spraying just headline, so a strobe, which is off-label once you get into that boot stage. But how that, once you get to flowering or in the boot, you can potentially elevate the mycotoxin compared to that non-sprayed control, which is really intriguing. There's been a little bit of data out of Canada to demonstrate something similar, but there's not enough to satisfy us, I guess.
Yeah, yeah. So you generally like in wheat, if you look at the labels, anything that contains a strobilurin always has a hard cutoff rated anthesis, so because of this exact problem.
Right.
Yeah. So,
Um,
what'd you learn here?
Well, so this was just trying to think about the trials that we have and the products that are labeled for mycotoxin suppression. Um, the so what we have here is the different modes of action. So we've got in orange the DMI, which have been a stalwart of mycotoxin management in wheat.
Hmm.
The blue are the SDHI. So this is really relatively new. Miravis Ace in wheat includes that pidiflumethafen in blue there. In this case, it's Miravis Neo for corn, and they do include a strobilurin which is shown in that yellow column. So just looking at what is available, I've just put in green there a couple of products that are available or are labeled rather. So Proline, so it's got a suppression of gibberella and ear rot. And then that Miravis Neo has a suppression of Fusarium ear rot as well.
Yep.
But otherwise, maybe they've got a label in wheat or something, but really nothing else in the corn market. So here's some data. This is actually, compiling 4 site years of data that we had. So 2 years, 2022 and 23, but two locations each of those years. So just pulling that data
Hmm.
together.
Yep.
So we did a fungicide efficacy trial. We didn't have a lot of leaf diseases as we were sort of initially hoping for on the tar spot front.
Hmm.
But really interestingly, when we looked at the mycotoxin, we see some things that really stand out here. So that orange bar is the non-treated check. Okay. And we've got zearalenone on that whole left graphic and deoxynivalenol or DON on that right graphic. And so what I think is interesting is that if we look at Proline in that pink or salmon color, we see a little bit of suppression, not statistically different, but a little bit of perhaps suppression of those mycotoxins. So that's one of those labeled products.
Yep.
Miravis Neo in that dark green, a similar response there too.
Yep.
But when we did a combination headline amp plus Proline, that saw the biggest drop in mycotoxin and some statistical separation, either with the untreated check or with a fungicide that just wasn't bringing any performance to it. I thought this was really intriguing, and so we actually set up with a corn disease working group that's really focused on grain corn, looking at some of these treatments nationally here. So we're doing that this year, and I'm really excited to see what data we get back from that. And we've built on this too here in our silage program and looked at that and other combinations of Proline with other chemistries to see if we can get any effect. I just want to make a quick note that headline AMP includes metconazole, which was a product used in wheat head scab management. And so maybe because we've got metconazole plus that Proline, basically a 2x system, basically, we perhaps getting that reduction in mycotoxin. I just thought it was really intriguing, and we're certainly trying to explore this further in our grain corn and our silage work too.
So is this that lower rate of headline amp, or is this the high rate, which I think is a 14
It,
ounce or 14.4 ounce rate or something like
That's
that?
a great call, Damon. This is at the slightly lower rate. So I
Yep.
think we went with 10 fluid ounce and then the 5.7, I
Yep.
believe, of Proline. That's right.
We've seen similar things with headline amp where we've had the higher rate of headline amp. We haven't
Mm,
spiked with Proline, but used the top end rate, and we get some decent suppression of deoxynivalenol, which is interesting because it is paired. That metconazole is paired with a strobilurin, right? So
right.
there are some interesting things that don't quite follow the wheat story, right? Where the
Hmm.
strobilurins may not be driving mycotoxin up, but maybe they're just not controlling it quite as well because you do have some other things here like Quiltic cell, which has a strobilurin, maybe not doing much in terms of control.
Right.
relative to the non-treated. Yeah. So it's really all about having that high level of DMI in there still, right, Marty?
Yeah, it certainly looks that way. I mean, I guess the only caveat to that is that Miravis Neo that has the pideflumethafen that we have
Yeah.
both seen, or at least that combination seems to do a pretty good job in wheat.
Yep.
And so yeah, that's right. I mean, there's so much to explore here, right?
I know. Yeah.
Yeah,
Yeah. Yeah.
yeah.
So, so yeah, with that, I think if you want to learn more about what we've been doing and mycotoxin management and silage management and that sort of thing, I think it'd be a good idea to check out the Crop Protection Network site. We got a lot of resources there, Marty, including some of the tools and things we talked about, the
hmm
forecasting tools and all that are all housed at the Crop Protection Network there. Anything you want to finish up with?
Just feedback is always welcome, right? You're always looking for feedback on your models, like good or bad. We want to hear it, and same with the Crop Protection Network. Is there some way that we can be better serving you, or the particular studies you want to see? Please let us know. We appreciate the feedback.
Yeah, I second that. And if you, especially with the forecasting tools, we've been through one season now there. There's actually down in the lower right-hand corner of those tools, there's actually a link there where you can give us online feedback. So if you want see some improvement or you have a suggestion, feel free to click on that link and send us something electronically.
Yep.